We're very happy to have Maria Arcila, Memorial Sloan Cancer Center, as the speaker today on this webinar. Maria is an anatomic and clinical pathologist with a subspecialty training in molecular genetic pathology and hematopathology. She currently serves as medical director of hematopathology and deputy chief for lab operations for the molecular diagnostics service at Memorial Sloan Kettering Cancer Center in New York. Maria received her medical degree from the University of Pittsburgh School of Medicine and completing her residency training at Magadan Army Medical Center and fellowships at Memorial Sloan Kettering Cancer Center. She has a special interest directly involved with the expansion, validation, and implementation of clinical molecular tests that are relevant to the management and treatment of patients with both solid tumors and hematologic malignancies. A fun side note, Maria is from Colombia and a bit of a foodie. Her care and attention to detail extends to food and coffee preparation. And her family owns a coffee plantation in Colombia where she receives coffee beans, roasts and grinds them, and prepares them in a very specific, careful way. So there's no Starbucks for Maria. And at this point, I'll turn it over to Maria. Thank you.
Wow, thank you. Well, yes, I do like good coffee. Well, so let's just go directly to this presentation. Thank you, everybody, for registering. If you have any questions, please feel free to ask them at the end or during the presentation. So today we'll be talking about NGS-based clonality testing, how to assess clonality status by next-generation sequencing, also the assessment of somatic hypermutation and monitoring minimal residual disease. The educational goals for this specific presentation will be to review the principles of clonality testing, discuss the role of next-generation sequencing in assessing clonality and somatic hypermutation, and also the role of next-generation sequencing in disease monitoring and minimal residual disease assessment. We will discuss along the way some of the bioinformatics software and the data analysis. And also, we, at the very end, will discuss a little more on why and when should we get next generation sequencing and when should you consider doing this type of testing, and also the benefits and the potential pitfalls of this kind of testing as well. So as you know, clonality testing is an integral part of the assessment of both B and T cell malignancies. The diagnosis of both B and T cell neoplasms can be greatly supported and facilitated by clonality testing. And depending on the experience of the hematopathologist and the type of the histopathologic request, approximately 25% of cases that go to an institution for histologic assessment and diagnosis would require clonality testing for diagnosis alone. Of the many different markers that can be used for clonality testing in suspected lymphoproliferations, immunoglobulin and the T cell receptor antigen and the T cell receptor are the rearrangements that stand out as most widely applied targets. Usually PCR-based analysis in the past 2 decades has been the gold standard method for assessment of clonality. And this specific kind of testing has some difficulties in terms of the assays are not extremely sensitive. So while we can use clonality testing to be able to diagnose disease at the time that you have active disease, and a lot of it, the use of clonality testing for monitoring of residual disease has been really very limited because of the intrinsic relatively low sensitivity of the routine standardized assays. To be able to talk about clonality testing, we really have to review some of the biology and understand how clonality testing has been designed and why the assays that we have work the way they work. So we know that in the normal physiologic state, both immunoglobulin and the T cell receptor undergoes rearrangements that are formed from very early stages in both B and T cell development throughout the development of both. there's this random coupling between different segments of the genes that results in the formation of a unique sequence in the DNA that encodes the actual antigen binding regions of both the immunoglobulin and the TCR chains. And as a consequence, each lymphocyte is going to bear a unique antigen receptor molecule on the membrane. And the chance that you're going to have two different lymphocytes coincidentally having exactly the same receptor with the same sequence is almost negligible. So identical rearrangements are not derived from multiple independent generated cells, but rather reflect the clonal nature of the involved cell population. So establishing the unique length or the sequence of that specific molecule will allow us to discriminate between monoclonality and polyclonality. So we usually say that when something is polyclonal, we consider this to be benign, but this is not always the case. And when we talk about monoclonal, we generally consider this to be neoplastic because we have one product that is completely overrepresented and generally coming from a single cell. Very briefly here, as I mentioned, these rearrangements will occur in both the B and the T cell receptors. So using the B cell receptor as a model in the B cell, for example, the changes again occur through several stages, and each stage represents a change in the genome content of the antibody loci. So in this diagram that you see here, just to remind you, this is the schematic representation of an immunoglobulin molecule where you have an antibody that is composed of two identical heavy chains and two identical light chains. And there are genes that specifically code for specific regions, and the regions that we usually think about when we talk about the immunoglobulin is the variable region, the diversity region, the joining region, and the constant region. And these are all important to recognize when we talk about clonality testing. So the process of rearrangement of these molecules happens in two locations in physiologic conditions and also in pathologic conditions. So you have an antigen-independent B cell differentiation, which happens in the bone marrow, and then you have an antigen-dependent B cell differentiation that happens in the periphery. When you're in the bone marrow, you have your stem cell that is going to go through several stages from pre-B to B1 to B2 to pre-B2 and the immature B cell. And then once this cell goes to the periphery, you have a transitional B cell that will either become a naive mature B cell that can either go through the germinal center and become a plasma cell or a memory B cell, or you may continue to have a naive mature B cell that stays outside of the germinal center. Through this entire process, you're going to have several changes in the immunoglobulin molecule, and the first thing that usually happens, as you can see here, is the DJ rearrangement of the regions, the diversity region and the joining region. Then immediately after that, you're going to have the rearrangement of the VDJ, and then after that, kappa and lambda as well. As the cell goes to the periphery, you're going to have additional changes that happen to these molecules, mainly somatic hypermutation and a class switch from IgM to IgE or IgG. And this happened now very, very late in the different stages of maturation. All right, so in clonality, when you do clonality testing, you are basically testing primarily the variable region. So this right here is the structure of the of an immunoglobulin chain, and this is actually the gene. So the gene is composed of different regions, as you can see here. You have the variable region, you have the diversity region, you have the joining region, and then the constant region. And then you have different exons that code for each specific one of those. You have many for the variable region, about 66. Many of these are non-functional, but about 50 of them are functional, and then you have the diversity region with about 27 coding exons, while the joining region is about 6 of them. In most cases, both physiologic and pathologic, in about 75 to 95% of the cases, the usage or the section that ends up being coded is within exon 1, 3, or 4. So the actual rearrangement of the gene happens by specific excisions of large parts of the gene, as you can see here, which form these circles that are called the BREC, or the B cell recombination excision circles. And these are completely different in every cell depending on what part is being rearranged. At the very end, once you get rid of all of your introns and your B cell recombination excision circles, you end up with a what's going to be your mature IgH mRNA. In addition to these changes that I just talked about, you also have additional insertions and deletions in the joining regions in between. And these are very, very small insertions or deletions that happen to give you more variability. And then once you have your mature mRNA, this is translated in what's going to be the actual immunoglobulin chain. And then you also have to transcribe your light chains, which form part of the mature molecule. So the resulting diversity in IgH, for example, or the TCR molecules after you have a lot of these changes, is actually very, very high. So through these gene segments that are being rearranged through the VDJ rearrangement, your combination diversity and your junctional diversity, you end up with a total diversity of about 1 times 10 to the 12 in both the IgH and the TCR as well. So this brings a lot of problems in terms of designing assays that specifically target these regions, because you have so many changes that then you have to design the assay very carefully to be able to target regions that are so completely different. And we're going to talk about a little bit more later on how those assays are designed. So in terms of the further modifications, since we're going to be talking about somatic hypermutation testing, once you have this molecule completely arranged, you're going to have additional changes that happen in the periphery. You have small changes in the variable regions you can see here, and these are your somatic hypermutation changes that increase the affinity of the molecule for a specific antigen. So you're going to have several changes that only happen in the germinal center. In addition to somatic hypermutation, you have changes in the constant domains, which is your class switching recombination that will change the effector function. All right, so let's talk about somatic hypermutation first. So somatic hypermutation happens, again, in the germinal center. You're going to have changes specifically in the bases, and the experimental evidence supports the view that the mechanism of somatic hypermutation involves the deamination of cytosine to uracil in DNA by an enzyme that is called activation-induced deaminase, which happens to live in the germinal center. So cytosine and guanine pairs are directly mutated to uracil and guanine mismatches because of the activity of these deaminase. The uracil bases are usually removed by the repair enzyme uracil DNA glycosylase, and then this actually calls error-prone DNA polymerases that are going to be recruited to fill in the gaps and create mutations. So when you have very rapid proliferation of different populations of B cells, and with the addition of all of these mutations that are happening at the same time, you're going to produce thousands and thousands of B cells that possess slightly different receptors in varying specificity for the antigen, from which the B cell with the highest affinity for the antigen can then be selected. How do we determine somatic hypermutation? So if you have the sequence of that final molecule, you are going to determine how different this molecule is from the germline molecule. So somatic hypermutation usually happens in the complementarity determining regions, and these are the preferred sequences for somatic hypermutation, but they can easily also happen in the framework regions, which are part of the variable region of immunoglobulin. And these, even though they're supposed to be very highly conserved regions, they also get hit by somatic hypermutation, making it difficult when you're designing an assay to have primers that anneal appropriately to the regions that have been highly somatically hypermutated. So if you have the germline sequence, then you can align the sequence that you have and determine how many of these mutations you have compared to that germline sequence and come up with a rate of mutation. If you have a mutation rate that is higher than 2% compared to the germline sequence, you call that case somatically hypermutated. All right, so PCR-based analysis of immunoglobulin and T-cell receptor rearrangements is the gold standard method for clonality testing and has been used for the past 2 decades. The analytical phase of the testing has been extensively validated and is largely standardized. So the initial multiplex PCR assays were designed by Biomed 2, now called the Euroclonality Consortium, and the primers have been optimized and the assays commercialized by Invivoscribe. This very, very optimized design allows a very high rate of detection of the most common B and T cell malignancies, regardless of all the changes that are happening to these molecules because of the optimized design. So in general, the capillary electrophoresis assays, for example, so you do PCR and then you do capillary electrophoresis, are the usual assays that are used across the country and across the world for testing B and T cell malignancies. When you have your gene, your IGH gene on chromosome 14, this is divided into specific sections, the FR1, FR2, and FR3, and in between your CDR regions, so which are your complementary determining regions. The framework regions, the 1, 2, and 3, is where the primers usually sit, because these are the regions that are less hit by somatic hypermutation. And this allows you to have a higher pickup rate for clonal rearrangements in neoplastic conditions. So when you organize these fragments by size, if you have a non-neoplastic condition, or you have a polyclonal distribution, you end up with something like this. You have fragments that are of different size that are evenly distributed in this Gaussian kind of curve. If you have something that is clonal, where every single cell has the same rearrangement, you're going to end up with a single clone that looks like this, and you don't have distribution that you have in here. And it's usually great if you have something that is distributed so nicely into a polyclonal, nicely Gaussian curve and a clonal single peak. But this is actually not the case for any of us that work in the diagnosis of B and T cell malignancies. And what you usually end up is with things like this, where you have a clonal peak that may be very prominent, but you could have a very high polyclonal background, or you end up with something like this, where you have just a few peaks and some peaks in the background, but you don't really know what exactly to call clonal. So the background knowledge and ample experience is really required for an accurate interpretation. It's not just a matter of calling clonal and not clonal because you see the peak. But there are also a lot of interpretation algorithms that exist that take into account the different peak heights and the peak ratios to define what is truly clonal. But then again, you use cutoff values and algorithms that sometimes create a false sense of accuracy and might even lead to false positives or false negatives in the interpretation because you're just interpreting a peak. So these assays that have been used now for many years are they provide a very simple process. They're robust and very rapid in their analysis. They usually require low DNA input, and they're relatively inexpensive. It successfully exploits the size and the overall composition of the rearranged fragments, and that the sensitivity is low, is about 5 to 10%. And then, but it's available to any institution and is used by most laboratories. The problem is that, this separates the PCR products by the length of the rearrangement and not by the unique sequence. And you may have fragments that are of exactly the same size, but they're not necessarily the same sequence. And the interpretation may be very subjective. And again, they have relatively low sensitivity. So of course, we haven't been able to use these assays for minimal residual disease testing because you just cannot get to the sensitivity that you need. So clonality assessment by next generation sequencing methods has marked advantages over the length-based analysis methods that I just showed you. It allows the identification of the full range of clonal populations and determines the specific DNA sequence of the clonal rearrangement. You can detect clonal events that are hidden in a polyclonal distribution, and you can easily track residual disease at very, very low level that is required for MRD assessment. Because the assay is made in such a way that you're determining the exact sequence, you can also examine somatic hypermutation as a prognostic marker for CLL or for hairy cell leukemia or for any of the diseases that somatic hypermutation has been shown to be important for. But beyond the clonal detection itself, you also learn a lot about the biology of the disease. You may identify both stable and dynamic aspects of the immune repertoire that differ during both normal and disease conditions, and you provide a very high resolution picture of the spectrum of immunity that is found in lymphoid malignancies. What we have found through instituting next generation sequencing at our institution is that we can actually define the initial behaviors of a clonal tumor population, and we can very easily see the suppression or the reemergence of different populations following treatment. So a lot of information coming your way, which we are actively using for decision making in many of the cases. So the NGS-based clonality testing can be done through several different methods. They usually utilize capture-based approaches, and they can be either in-house developed or commercially available. We specifically use the Invivoscribe products, the LymphoTrack assays, which are commercially available, and they enable the assessment of IgH, IgKappa, TCR gamma, and TCR beta for the clinical cases. It also uses leading next generation sequencing platforms. Either you can use Illumina or Ion Torrent, and it has optimized multiplex PCR master mixes similar to what you would use for the fragment analysis with primers with platform specific adapters. There is specific tracking with ID tags for single step workflow, and also comes with a comprehensive bioinformatics package where you can look at the DNA sequence and visualize the clonal prevalence as well as the VJ family identity for each of the rearrangements. So let's go briefly on how the assay is designed. So we before went over the structure of the gene, and we said that there are CDR regions in between that get hit predominantly by somatic hypermutation, and then you have the framework regions that are not hit so much. So then the primers for this assay are going to be designed specifically for these regions. In addition to these primers that are included here and also included for the CE assays, you also have the addition of lead primers, which can detect clonal rearrangements in case you have, well, number one, a lot of somatic hypermutation in this region, but also allows you to see the entire variable region of the gene for somatic hypermutation assessment. The gamma, the TRG gene assessment has primers that targets both the V and the J the same way that the CE assay would do. So, well, one question is always, how do you establish the diagnostic clone? So with this assay, I will show you some of the software output. When you have a case that you are running the lymphotract assay, you end up after sequencing with a screen that looks something like this. So the first tab in here, and this comes in Excel format after you do your analysis. You have a read summary tab. You also have a merge read summary. You have VJ, sequence, frequency graph, and you have other information here that give you absolutely everything that you need to be able to analyze the case. It gives you the sequence, the specific sequence of every single of the top 200 rearrangements.
Maria, just to interrupt, I hate to do so, but we're not actually seeing anything underneath on the presentation right now.
Here it is.
Oh, there it
really?
is. Okay. Yeah.
Okay.
It just came up.
It just appeared. Oh, okay, good. So what you see on the first screen when you analyze one of these assays is, again, the summary of the top 200 reads. It gives you the specific sequence of every single one of the rearrangements. It organizes it by the length and the prevalence. So you can, on your second tab, you actually have the merge read summary, which is basically you may have differences in these clonal sequences by only one base pair or two base pairs, which are then collapsed into a single read so that it's easier for you to analyze your samples. So once you collapse all of your reads, you have a summary of the most pertinent reads in here so that you can analyze your case as being clonal or not clonal. So in this case, for example, your top read is 82%. This is a case that would be called a clonal population. It also gives you the information on the mutation rate, and it tells you whether that rearrangement is in frame or not. You can click on the specific sequence so that you can see the entire sequence, as you can see here, and this is the sequence that you would use later on to be able to follow up that same clone in future samples. When you click on the sequence frequency graph, this is what you actually see. So you can easily see that the vast majority of the sequences that are in this specific sample share exactly the same clonal sequence. So it is a glance to be able to say whether you have a clone or not a clone. And it also gives you the visualization based on usage, depending on the percent of the total reads. So this is what a clonal sample would actually look like. For comparison, this is what the fragment analysis looks for the sample. So the actual interpretation is very, very similar once you look at the graph. A polyclonal software output would look something more like this, where you actually have your cumulative reads and your merge reads. Everything in here is just below a 0.04%, and all of them at around the same frequency. So there's nothing that stands out as being a dominant sequence. So this is polyclonal with the distribution based on usage that you can see here and the comparison to the fragment analysis in here. So we have our own software where we can actually organize the sequences based on size. So you can actually see the comparison here. So this is what you would expect if you have a polyclonal sample, where each of these different peaks actually have multiple different clones of different distribution that you can see in different colors in here. So what I actually wanted to show you here is that if you're tracking a clone in a polyclonal sample that you already know what the primary clone was at the time of diagnosis, you can use the MRD data analysis tools that are also provided, where you can search for the specific sequence, and the software will analyze it to tell you how many of those sequences actually match to the sequence that is diagnostic for the sample. It gives you the exact match with exactly the same sequence, and it also gives you the frequency if you have either one base mismatch or two mismatches to allow you for any type of error during sequencing. So one of the things during our validation of this assay was, of course, figuring out what the sensitivity for the assay was and whether this was actually more sensitive than our prior CE assays. What you can see here is a dilution study for the IGH assay. This is a 5% dilution, your 1% dilution, your 0.1, and the 0.01, and the comparison sensitivity study for the fragment analysis. At 5%, in the fragment analysis, you see a clonal peak in a polyclonal background, and as you sequentially go through your dilution, you completely lose that peak, and you can no longer make the diagnosis of clonality. With the next generation sequencing assay, because you are looking at the clone not by size but the actual sequence, you can easily tell that you actually have a clone, and you can go as low in here, for example, as 0.1% for the diagnostic sequence. The inter-assay and intra-assay reproducibility for both the IGH and the TCR for us was very good. This is an example of what we did for reproducibility for the IGH assay. And so it is expected with the different repeats to have certain variability on the percentages of each sequence that you see here. For example, is a biclonal sample, actually a biallelic sample, where you have a clone that is at 27%, and as you repeated both in the inter-assay and intra-assay reproducibility studies, the variability between one run or the other runs is actually very low. One of the important questions that I get a lot of the times is, how do you actually interpret clonality, and especially in very low tumor samples? What is the cutoff that we use? So our criteria for calling a clone is actually very stringent, because we use the assay primarily not necessarily to call a clone at the time of diagnosis, but to be able to use that clone later on for monitoring disease. So this, for example, is a sample that if you are looking only at the graph, you would be very, very tempted to actually make a diagnosis of clonality, because you have a very prominent clone with exactly the same sequence, and then a polyclonal background that is well below what your peak is here. This actually does happen to be the clone in this case, but when you actually look at the summary of the percentages of this clone in this specific sample, it's actually very, very low. It's only 0.54%, as you can see here. And even though it's so much higher than the background, we wouldn't actually make a call of clonality in this case. We have the rules that we have for diagnosis at the time that we see the first sample is that the top clone or the top two reads have to be more than 2.5%. And then more than 5% the percent reads of the fourth most common sequence. So the background, you look at what your background is, and you decide, and that has to be 5 times less than the clone that you're calling. We're also very stringent in the number of reads that we have. We have a lot of samples where we get very little DNA, especially for the samples that are paraffin embedded. But we do require that at least we have 50,000 reads when we run the sample to actually call this clonal, or we would repeat the sample a second time to be able to say this clone is reproducible if we have a low number of reads. So my recommendations when running an assay and calling a clone, if you're going to be going beyond the clonality but actually following that clone later on, is that it is really extremely important to standardize the DNA input and the protocols, and you have to set very stringent criteria for clonality calling for the diagnostic sample when you get that first sample. I am usually very aware of what sample is a post-treatment sample with very low level of disease for initial clonality assessment. And you really shouldn't be making a diagnosis of clonality in these samples for an initial clone. You have to be aware of the low template samples because, well, number one, they could give you a pseudoclone, and they really may require duplicate testing by this assay. And again, the interpretation, as with any other assay, it has to be made in the context of the disease that you're seeing and any other ancillary testing. And if it really doesn't make sense for the disease that you're looking at, you shouldn't be calling the clone to begin with. So one example of calling a clone that is actually not a clone, even though it met all of the criteria for a clone by our assay, was one example from this year. This is a patient who came in with a history of follicular lymphoma. This is a post-treatment sample in which, at the time that we did the testing, we didn't know what the disease status on the patient was. We found out later on that even though the sample was submitted as an initial characterization of the clone, the case actually had absolutely no morphologic disease. We did make the diagnosis of a clone, as this was about 70% of all of the other little sequences that you actually see here, and very, very, very prominent, way more than 70 or 80% of the other sequences that you see. However, later on, because the sample had no morphologic disease, we did request that they sent us slides of the initial sample, and this is the retrospective sequencing of the FFPE tissue from the diagnostic lymph node. And this is actually a completely different clone. So if we had followed this patient with this clone, we would have been following the completely wrong clone for this patient. That was not the one that was associated with the disease. How do these assays actually compare to the CE assays? And what is the extent of testing that you actually need to do? So I am borrowing this table from what was published by the Biomed 2 concerted action in 2007. There are samples that are just exceedingly difficult to show that you have a clonal population. So in this case, for example, they make a comparison here of what type of clonality you would get if you were running both FR1, or actually FR1, FR2, and FR3 primers, and whether you are doing all of the FR1, 2, and 3 plus IJ kappa. So there are samples, and usually these are either the ones that derive from germinal center or post-germinal center, that even if you do all of the testing, you are never going to get to 100% using the CE primers. So what happens with the primers by next generation sequencing, and how far can you actually go for a call of clonality? So again, here highlighting the most difficult samples that we find: follicular lymphomas, diffuse large B-cell lymphomas. If you only run FR1, FR2, and FR3, you get to about 80%. And if you add kappa, you can get to 100% in some of them, but some others you really cannot get to that level. So this is a summary of our first 500 cases, and whether we actually tested them with FR1 or a combination of FR1 and leader, or all of the primers, FR1, FR2, FR3 plus kappa. So what you can see here is that this is very similar to what you actually see with the CE assays. Follicular lymphomas are notoriously difficult kind of samples. So are plasma cell neoplasms and lymphoplasmacytic lymphomas, that if you only did FR1 primers, you would only get to about 60, 65%. So these are the samples that you actually have to do the whole battery of testing. But the vast majority of cases, you can get to very close to 90% if you only do a combination of FR1 primers plus the leader primer. There are only some cases where you're going to have to do both the FR2, FR3, and kappa to be able to get to 100% or close to 100%. And most of these are going to be your plasma cell neoplasms, which again are just notoriously difficult to show clonality. All right, so moving on to somatic hypermutation assessment. The somatic hypermutation assessment is really greatly facilitated by next-generation sequencing. Not only is it much easier to do because you don't have to go through the very extensive process that it actually takes to do somatic hypermutation if you use other methods, but it also demonstrates much higher sensitivity than the traditional methods because the assay itself is just more sensitive. So you can go below 5% to be able to capture some of those low tumor burden samples. But in addition to being easier and also giving you higher sensitivity, you also can capture multidimensional clinical data with a single assay. You can simultaneously look for the specific sequence. You can gather whether something is productive or unproductive, and you also have the concurrent determination of somatic hypermutation status. You can also easily characterize multiple clones when they are present, and it is actually known now that many of these cases have more than one clone that you may want to monitor later on during the disease process of the patient. So traditional clonality assays, just to review very briefly, incorporate not only the clonality testing itself, but then also you have to take genomic or cDNA as template, and then you have to clone it into a plasmid and then align this to be able to calculate the discordance of the sequence from the germline and calculate the somatic hypermutation rate. In our institution, for example, we didn't actually do this test. We used to send this out to another lab because it was just very difficult to actually do this and incorporate this in our workflows. You actually have to sequence, and reading sequence of rearrangement is actually extremely difficult. So it wasn't something that we actually wanted to do, and there were labs that were doing this. We brought, obviously, the test in-house as soon as we started doing next-generation sequencing, and it's something that you can do very seamlessly with this type of assay. So this is, for example, a case which I specifically selected because it was a very interesting case. This is a case where you're looking for a single clone. This is the fragment analysis. And you can see the clone here. Maybe there is a tiny little clone here that is a minor clone, but doesn't actually make the criteria for clonality. What you see here is that the clone is about 266 base pairs, and you can see the usage, the V and the J usage, and the total read. So what you actually have here is actually 3 clones. And the top clone is hypermutated. So the mutation rate is more than 11%. So that makes it easily, this is a hypermutated case. There's no question about it. This case was specifically tested with the FR1 primers. So when you take a look at your second clone, which is also very clonal, is 26% of all the clonal sequences, the mutation rate is 1.76, so it doesn't actually make that cutoff of 2%. And then you have a third clone in here, which is much smaller, which is actually not a third clone, but it's actually the same clone. This would be merged together. You don't really know whether it is mutated or not mutated because one of them is 1.76, the other one is 3.66. So this would be mutated, this would not be mutated. So the question is, what do you do with this kind of samples? So we generally test for somatic hypermutation with only FR1 primers. And we have found that if we do this, this takes care of the vast majority of cases. Something that is mutated is usually mutated and is well beyond 2%. But there are these few cases that are right in the borderline where you may have to do additional testing just to compare. So these would be the kind of sample that we would also do with the leader primer so that you can capture the entire variable region. So when you do that, and we actually haven't found that in any of the cases we have tested with the leader that we can get something to be not mutated to mutated, except on very, very special cases. So this case actually, when retested, didn't make the cutoff for hypermutation, but we do recommend that when you have these numbers between 1.75 and 2, that they get retested with the leader primer. You can actually see the sequence alignment here for these two sequences. And you can see that these two sequences, even though they are exactly the same length, they do have different usage and they have a different sequence itself. So we went through our, an entire validation again on many CLL samples, and we have been using the assay for about 2 years. We use the same rate of somatic hypermutation that is used for other assays as 2%. As the cutoff for calling something somatically hypermutated. We did do a comparison as well with a reference lab with other methods of about 50 specimens. All of them were completely concordant, and the assay itself has very, very good inter and intra-assay reproducibility and precision. But we do see also identical mutation rates in multiple repeats. We see as well, as I said, that the mutation rate with both FR1 and leader for most of the samples is going to be exactly the same. But again, we do recommend that if something lands in about 1.75 or 2%, that maybe that should be retested. All right, so in the next few minutes, we will talk about disease monitoring. So this is, we do test every case that comes into our institution with this assay to characterize the clone, and then we follow the patient later on with the assay to see if we can pick up the clone. So, so this monitoring is really one of the main advantages of next generation sequencing. So following the initial characterization of the disease-associated clone, it is possible to track the clone in subsequent samples at levels that are beyond what is allowed by flow cytometry, but the DNA input is absolutely critical to do the MRD testing. So here, for example, if we took a theoretical yield of 6.5 picograms of DNA per cell, if we wanted to test something with about 100,000 molecule equivalents, we would have to test about 700 nanograms. So the issue is how far can you actually go and how many replicates would you actually have to test to be able to get to the sensitivity of either 1 × 10⁻⁴, 1 × 10⁻⁵, or 1 × 10⁻�6, which is usually the number that gets quoted for any minimal residual disease assay. So this graph that you see here specifically depicts the confidence level of a negative sample at 1 × 10⁻⁴. And this again this is a theoretical plot that is driven by statistical modeling. If you were to test a single sample with only, say, for example, 500 nanograms or 1 microgram, you would have, with only that, you would have a 95% confidence that that a sample that is negative is truly negative, 95%, 95% with 95% confidence. So if you were going to go beyond 1 × 10⁴, you actually need a lot more input. The question is, if you have to do multiple replicates of 1 microgram to get to a sensitivity of 1 × 10⁻⁵ or 1 × 10⁻�6, you would be ending up with about 7 different replicates on each individual patient, which would constitute an individual run for that patient. And this is not something that you can actually do clinically. So if we were looking again at what our theoretical is going to be, if you really wanted to hit that 1 × 10⁻⁶, with 1 microgram, you would have to do, if you did 2 replicates, for example, you would get to that confidence only 14% of the time. But if you were going to go beyond that with more replicates, you would be able to get that to the 1 × 10⁻⁶. This is again, however, what you're looking at if you were hoping to get, 5 reads of a target sequence with 95% probability. So and this is theoretical. So if you actually wanted to go if you actually want to do this test in a clinical setting, you cannot actually do this, as I said. So we went on for our own validation to only test anywhere between 2 to 4 replicates and see where this actually got us. So this is the summary of what our initial dilution study for our sensitivity assessment and our validation of residual disease. So what you see here is actually a dilution of a clonal sample into a non-clonal sample that is diluted from 1% or 1 in 100 to 1 in a million for the 1 times 10 to the minus 6 sensitivity. And we use, again, a cell line, and in here, 4 different replicates, which happen to be at 500 nanograms each. So in practice, what you actually see is that you can very easily get to this 1 times 10 to the minus 4, even if you only do a 500 nanogram input. You can even see the sequence very easily, a positive, you can very easily detect positives, and you may not have 100% or 95% confidence, but you can often see the positives. And here, for example, this is the 1 in 100,000, and you can, in the 4 replicates of a 500 nanogram, with a 500 nanogram input, you can see it in every single one of the replicates. When you go beyond that, that's where the problem is. So, we can confidently say, based on our assay and our validation, that we can get to 1 times 10 to the minus 5. But with this kind of input, we would not be able to get to 1 times 10 to the minus 6. And again, what you would require, or what we require to call something positive, is about 5 to 10 reads in every single one in the combination of runs. So we usually run 2 runs of 500 or 1 microgram. And if we see the sequence in both of the replicates, then we call the case positive. If it is negative, we repeat it. And then depending on how many you actually have that are positive, we will call positive. So the bottom line is that getting to a sensitivity, if you were going to be completely strict about your input of 1 times 10 to the minus 6 is really not possible, given the amount of work that it would require to do that. But we do know that we get to the 1 times 10 to the minus 5. This is a comparison of next generation sequencing with flow cytometry from some of the samples that we did during our validation. An important aspect in here is that even when your flow is really, really, really good, and I think that we have extremely good flow cytometry at our institution, in some disease processes, the next generation sequencing is actually much better than flow cytometry. And what you can see here is for samples that are that are acute lymphoblastic leukemias, mature B-cell lymphomas, and mature T-cell lymphomas. When you test the sample by flow cytometry, many of these are going to be positive, but if you test it by next generation sequencing, you're going to have at least 20% more cases that are actually picked up because you're looking for the specific sequence. The same again is true for the mature B-cell lymphomas and even more for the mature T-cell lymphomas because some of these are very difficult to actually diagnose with flow cytometry. So by far, the next generation sequencing is going to be better. With plasma cell neoplasms, that actually doesn't seem to be the case, and flow cytometry does pick a few more samples. These are all samples that are at the very, very, very low level and really pushing the 1 times 10 to the minus 6 level. So in here, we only had a few samples, but given that plasma cell neoplasms is one of the things that we have a very high interest in defining at our institution, we went and looked at a few more cases. So it so happens that at the end, the detection of plasma cell neoplasms, of minimal residual disease in plasma cell neoplasms is very, very similar for both the flow cytometry and the next generation sequencing. And again, the few cases that we are missing are at this very, very, very low levels. And these are the responsible ones for the discrepant cases that we have had in our series. We do, by looking at the distribution of all of the very, very low level samples, that we are detecting low-level disease compared with the flow cytometry at these very low levels, close to 1 times 10 to the minus 6, even though we're not using the very, very high input that is required to really say that you have 100% or 95% confidence that you're going to detect it. So in the next few slides, I just want to show you some examples of the benefits of next-generation sequencing testing. Based on our experience, you can detect a diagnostic clonal sequence at very, very high sensitivity and specificity, of course. But you have this very high resolution picture of the spectrum of the immunity found in that lymphoid malignancy that is well beyond anything else that you can possibly get with any other assay. But more than anything else, you can actually define the behaviors of the clonal tumor populations because you're seeing the suppression or the reemergence of different populations. So here, for example, is a case of a 2-year-old male with BALL. This is a diagnostic sample with an ATV6 RUNX1 fusion, where we characterize the initial clone. When you look at this later on in the monitoring sample, the morphology on the monitoring sample was completely negative, the flow was completely negative, and the fish was completely negative. However, we picked up a sequence at 2.5% of the clonal, of the rearranged reads with very, very high read support. This patient, even though was negative by everything else, we went ahead and did the qPCR testing, and this patient does, in fact, have disease, and the patient was monitored as having disease based on the next generation sequencing assay. And this is a screenshot on how the MRD data analysis tool looks like for this specific patient. The next case is a 52-year-old male, also with BALL, where a diagnostic sample shows that this patient actually has 2 clones, which you can see here. When you monitor this patient on the next sample, on the relapse after 1 year, has only 1 clone, which is actually the minor clone that you saw initially, and this actually became the resistant clone, while the other clone was only seen at very, very low level. The subsequent samples, 5 different samples that we saw on this patient, only harbored the resistant clone, and the initial clone was completely lost after treatment. In the pre-transplant samples, even though any other assay would be completely negative, except for the flow that was that had a very, very low positivity, we can still very easily track the clone by this specific sequence that we're seeing. This is another case where you can actually see the emergence of new clones. This is the case at the time of diagnosis, and you can actually see the fragment analysis at the same time. And this is the relapse sample where you actually have an additional clone that you didn't see before that is emerging, that you can then easily track the 3 independent clones in future samples. And finally, I think I have two more slides. This is, for example, another case that was very interesting. This was a right parotid lesion that had 2 different clones. By fragment analysis, it looked like a single clone, but it's actually 2 clones with 2 completely different sequences, but exactly the same length. This is a patient that in the staging marrow, the patient didn't seem like the marrow had exactly the same clone as the parotid, but we, by fragment analysis, couldn't really say that that was the case because we couldn't truly call this clonal, except that there were 2 prominent peaks. We did, however, take this clone and we followed the patient with this specific clone, and we compared it to the initial parotid clone, and it so happens that the clone that was seen in the bone marrow subsequently in the disease actually showed that the T-cell lymphoma that this patient had compared to the parotid lesion was actually a completely different clone that was not initially seen in the parotid sample. And then finally, this assay actually makes it very easy to follow multiple clones depending on what disease they have. This one is a case that by flow cytometry, you could easily see 3 abnormal populations. One was a hairy cell leukemia that was lambda. a CLL population that was also lambda, and then a CD5 positive B-cell lymphoma that was kappa. You can easily see the 3 different clones in here. These the same usage, but different sequence, and then the 3rd clone right here. You may need for the initial assessment to be able to discriminate between the 3 clones. Sorting would be easier, and then sequencing the sorted sample, and then doing the follow-up of the patient with next generation sequencing only, where you can actually track each individual clone. So far in our experience, we have done about 1,200 samples. We're following about 800 patients. And what we do see is that the implementation, this is very feasible to implement, but you may have to do it in a stepwise fashion. In our case, for example, we decided that a lot of our cases need fragment analysis because you need a rapid diagnosis. But immediately after we received the diagnostic sample, we screen with the capillary electrophoresis assay to be able to give the clinicians a rapid diagnosis in the cases that they need. And then we do the characterization by lymphotract, and we do it with one type of primer or several primers, depending on the specific disease process. For cases that are not clonally, that do not have a clonal rearrangement by fragment analysis, we always review for the content, the tumor content, and then if the tumor content is between 5 and 10%, we retest them with the next generation sequencing assay because the assay is more sensitive, so it makes sense to characterize at that point. We do not characterize anything that is below 5% as a first sample, just because we don't want to run into issues with pseudoclonality or calling something just because we had a very low template. We have created, to be able to handle so many samples, we created our own portal where we can keep track of each individual clone, where we can actually do the MRD testing. But this is something that requires a lot of bioinformatics, support. This was a portal that was developed by Mustafa Syed at our institution, and it allows us to do many samples at the same time, and it allows us to track the clones very easily and generate reports very easily as well. So lastly here, when should you really consider clonality testing by next generation sequencing? So if your idea is to just use the assay as a diagnostic assay and rapid turnaround time is required, you should only do the fragment analysis assay. However, if your intent is to follow that patient later on and do any type of monitoring, you should be doing next generation sequencing in all of these samples to be able to allow that. Important things for monitoring use, you should consider the sensitivity and the feasibility to meet the expected turnaround time that is required, and also consider what kind of ancillary methods do you have in your institution if you need a rapid turnaround time. Should you be doing qPCR or flow in addition to doing the next generation sequencing? But this is a decision that is purely at the institutional level. However, it is important to consider that in certain disease processes, such as in T-cell malignancies, in BALL, and some of the low-grade B-cell lymphomas, the next-generation sequencing is really superior to flow cytometry. The potential pitfalls are actually the same as they are for the CE and a few more, but the important thing is that this is new technology that is not necessarily readily available in all labs at this point. The analytical phase is still without extensive validation and standardization. And for minimal residual disease assay, we really need to standardize testing to be able to say that these assays are going to be used across the board and to be able to compare results from one institution to the other. Again, there are no established protocols or guidelines, and so there are still a lot of unresolved issues on how to best quantify the degree of residual disease, whether we use a spike-in, whether you use an internal control that is specific to the patient, similar to qPCR. And there's also it's difficult to compare the data from flow cytometry and qPCR and digital PCR. So a lot of standardization still needs to happen. So in conclusion, clonality testing does provide very distinct advantages over other assays. It efficiently detects IgH, IgKappa, TCR gamma, and beta rearrangements using a very streamlined approach and a single workflow that results in very highly reproducible results and provides a more objective way to determine clonality. But more than anything else, it enables the assessment of the patient for monitoring of disease later on. It defines behaviors of the clonal population, suppression and reemergence following treatment in a way that no other assay can do. And compared to flow cytometry, it does provide higher detection of residual disease. In many of the diseases, it does have, in our experience, similar capabilities as flow cytometry for plasma cell neoplasms. But finally, further studies are really needed to define clinically significant levels of minimal residual disease, establishing guidelines that are based on longitudinal follow-up of the patient, and the outcomes are really necessary.
Maria, thank you, and acknowledgments are very important here. I appreciate.
Absolutely.
A very great team you have there.
I have an excellent team.
Nobody's going to dispute that. Hey, we have a couple of questions. First, if you were going to pick among the assays for tracking lymphoproliferative malignancies, how would you do it? Would you do framework? You said framework 1 and leader, I know. And what other of the targeted areas would you include, say, for IgH?
Well, for most of the samples that we have tested, we have been able to pick up with the clone with FR1 or leader. So if those two are the ones that are rearranged up front, we will route that patient to be followed with those specific two, because they're, number one, the ones that we have the most experience, and the ones that we see that are more seem to be most reliable. The FR2 and FR3 we do only occasionally in some of the patients. If we just haven't been able to find anything else, we'll do FR2 and FR3. I wouldn't be able to say much about Kappa right now because we're just validating it. So I really don't have experience on how to follow these patients with the Kappa specific assay. With the gamma, there's only one assay, so that's what we use for T-cell malignancies.
Yeah, and it's important to point out that even alpha beta T cells harbor generally biallelic TCR gamma rearrangements. Even if they go on to be alpha beta T cells, they still can be diagnosed, and malignancies can be diagnosed with the TRG assay. Answering another question that came in, the question was sensitivity between FFPE, peripheral blood, and bone marrow. What's your experience with those different specimen types?
So we have actually had very, very good luck with our paraffin embedded tissue, and this has been a question always. Are you able to work with paraffin embedded tissue? So I think that when we have done the characterization itself, I don't see any difference between the blood, bone marrow, and the paraffin in terms of performance, as long as you have a good sample. If you have a very, very scant sample with formalin-fixed paraffin embedded tissue, if I have any questions on the number of reads or the predominance of a clone, I would run a second time, which may not necessarily be the case for the for the bone marrow and the blood samples. So again, you just have to consider what is the amount of template that you have. And then for minimal residual disease, I have only used bone marrow samples and blood samples because they provide the DNA that I require to be able to use them for minimal residual disease assessment.
Okay, fair enough. And just to build on that a little bit, all of our assays, we wanted to initially design and do feasibility studies for picking the primer sequences to show that they're robust and reliable with the most challenging of samples. So we initially do assessment with FFPE specimens. And then only then do we move on to peripheral blood and bone marrow, because we realize those are just like falling off a log if you can get FFPE signal. To answer one of the questions, to answer one of the questions from, go ahead.
No, go ahead.
Okay, and then to answer one of the questions that came in from one of our participants, are the framework 1, 2, 3 leader, you know, when you combine assays on a chip, you can put all of these assays, frankly, at the same time on a single chip, so you can run whatever comes in the door that day to maximize turnaround times and minimize costs. The question was, are the analyses done comprehensively and together, or are they individually done? And our software does analysis for each of the target regions independently, along with the sequence ID for the specimen with the controls. So you look at the data and the outputs for each individual marker independently, and then you can assess whether, you know, what you want to combine for the final report or the final assessment. And then any other things that you would that come to mind for kind of comprehensive wrap-up of this webinar? Excellent webinar, by the way.
No, I think that the most important thing really is your validation and your monitoring of the assay, because there's a new assay. Every institution is going to have their own issues. From our end, our most important thing was to, number one, make sure that the performance of the assay was similar for both the paraffin embedded tissue for very low template samples, and ensure that what we were calling was actually a clone. And we have adjusted our interpretation rules to ensure that without a doubt that what we're calling the clone is the clone. I think, again, as I just said in one of the slides, it is very important that at the time that you determine what your clone is, that you have ample disease and you know exactly that this is your disease sample and this is what you're going to be tracking later on. Because if you do your characterization in the context of very little disease, you're going to have a high chance that what you're going to call a clone is not necessarily the clone that you're looking for, that what you actually have is just low template and you end up with a pseudoclone. But this is the same as it is for any other clonality assessment. So the fact that you have an assay that is more sensitive doesn't mean that for the characterization of the clone, that what you're going to do is go ahead and characterize something that morphologically may not have disease or has very little disease. I think that's my own caveat, and I do think that it's important to do when you're going to use the assay for monitoring of patients.
Okay, no, fair enough. And one of the questions came into to kind of the practical ramifications and the cost considerations of capillary electrophoresis versus NGS-based assays. As you indicated, the turnaround time for capillary can be very fast and inexpensive. But what we're finding also, one of the things that we mitigated was to be able to combine multiple assays at the same time. And you can really significantly reduce the cost of running NGS assays if you combine multiple specimens at the same time with the controls and run them all together. The costs approach the cost of running capillary assays. And so that's helpful.
Yeah, and I just think that the cost is significantly reduced in terms of not having, you know, you're not employing as many techs because you're consolidating everything into a single assay. And if you streamline your effort, you're actually not only able to save money on the actual running of the test, but also the time and the difficulties of running so many replicates, you know, tube A, tube B, tube C on IGH, and then beta and gamma. It's difficult. So we do envision that at some point we will stop doing the capillary electrophoresis. And I think that the only issue right now is really the turnaround time that we need for certain samples.
No, fair enough. And also, you know, as you indicate, you completely get rid of the somatic hypermutation. We have to identify the clone capillary, then you have to cut it out. You have to then isolate it, send it off for sequencing. And then that sequencing is only sensitive to about 10%, which makes it very difficult to make those calls. On one of the other things I wanted to mention was the MRD testing. We're trying to get international harmonization around the way that MRD is done so that, you know, we can have consensus around the metrics across institutions. So we're aiming, as you pointed out, you do, I think, 4 times 500 nanograms in the example you gave. We're suggesting about 700 nanograms is sufficient for most calls on minimal residual disease testing. But I think, as you indicated, every institution has to come up with criteria that they find important and appropriate for their institution and also their clinical practice. At this point, I think we've gone over in a very good way. I'd really like to thank Maria. I think I speak for everyone. We thank Maria Arcila and her excellent team for this wonderful webinar.